Innovations, Bangalore, and visiting scientist at Nairo Research, India, and also formerly at Marie Curie Research Fellow at Molcod Limited, Estonia, the country known for innovations. He carried out his PhD at University of Florida with Professor Katrzinski and Professor Carlson, University of Tartu. He holds 40 research papers in peer-reviewed and international journals, a US patent to his credit, and he has co-authored a book chapter on mosquito repellents. His expertise is in the area of computational chemistry, reaction mechanisms, QSPR, material informatics using ML. And apart from these scientific positions, he is also mentoring at Triple IT Kottayam and Government of India Niti Ayog's flagship scheme, which is Atal Tinkering Labs, at these schools in Delhi and around and in India, and is also the advisor for research at VIT Vellore. So now I invite Dr. Pillai for delivering his talk entitled Explore Bioactive Conformation Using Docking in CSAR. And before I hand it over to him, I would like to give acknowledgement to the DBT Star College scheme through which we have purchased this software from these Astra Innovations. Welcome, Dr. Pillai. Over to you for the talk.
Thank you so much, Madam, for the kind introduction. And also thank you other organizers, including Dr. Vandana and also ma'am. So it's a great honor that to address a large audience. As yesterday, Dr. Vandana was mentioning, even though during pandemic, we have all other hurdles, but to get a larger audience, online is the best mode. So around 146 participants are already here. Again, I can see that yesterday Dr. Hassan has mostly covered the sequence, collecting or taking PDB data, protein data bank, homology modeling, and also a little bit on the Autodoc Vina and also his front-end software, which helps you to run something similar to Autodoc Vina and preparation of the files and analysis. But today we wanted to see something on the same line, but on a different perspective. So let me share my screen. And all of you, please confirm that whether you will be able to see my screen.
Yes, sir, we can see your screen.
Great. So give me a second. Let me make some settings so that we will be fine. Okay. Okay, great. Now, before we start, I want this session to be really interactive. I can understand if someone is having difficulties on putting your audio or mic on, you can always go for a chat window. But before I start, it's better for me to understand the participants. So today, do we have most of the students from biotechnology or bioinformatics or pharmacy or microbiology, zoology, botany, just to have an understanding? I thought of doing a poll, but yeah, too much audience, so it will be a bit difficult. So that's why I kept quiet. So it would be nice if you can give an understanding on that. If the organizers or the participants, either way.
Mostly we have biology students.
Okay.
Dr. Girinath, some are from bioinformatics. There are many who are actually actively doing docking, especially from the lab of Dr. Hassan. Okay. And rest, there are a lot of microbiologists over here, but okay, they have less exposure to bioinformatics as such.
Exactly. That's exactly I wanted to understand, because I shouldn't go the way that every audience is having understanding of this, and I shouldn't go that way. That's why I thought to ask this question. So that I can also see in the chat, many of you are from zoology, microbiology. So I make sure that my discussions and explanations go in that perspective too. And now the second question comes is that how many of you think docking is just a job of getting score and selecting compound for your bioactivity? And how many of you does not think that it is not that the way, just depending on numbers, we can choose the compounds? For the first one, you can type 1. For the second one, you can type 2. So one was you believe that just based on docking numbers and energies, you can choose the compounds and go for choosing that compounds for your wet lab or molecular level studies. And the second option is that the numbers are not enough for you to make a decision. This is my single or 2 compounds that I have to take it forward. And most of you, I can see you're putting number 2 as the way I asked the question. But there are chances for 1 also, which I will be discussing more in my slides. And finally, one more question. Many of you would have worked on Autodoc and many other tools. As Dr. Hassan yesterday mentioned clearly, every software has its own advantages and demerits. One should understand what is that is fitting for you. Now, coming to the understanding of what is the difference between a paid software and a free software. If someone knows, you can unmute and tell rather than putting in the chat. What is the difference between paid and unpaid softwares or free softwares?
Paid ones are more user friendly.
Okay. That's true. Very true. Some extra features on paid. Yes, to some extent it is true.
Yes, sir. Like in free available softwares, like we have to do all the things like command line. We have to put the command there, but in the paid software, we just have to click the option and the whole procedure, it will be going to do it by its own. We have to do nothing, just to analyze the data, which is
Okay.
also in the paid software. It makes easy to use. Like.
Okay, you're right. Any other perspectives? You're still not come to the one which I'm going to tell you. Namrata, you can publish using free software, no problem. Only limited features can be used on paid, and to access to all features, you need to pay for it. Yes, of course. Paid software, you have to pay. Without that, it will not work. Doesn't fulfill need, editability of the software. Okay. Okay, I'm not taking too much time. Let me go to the point. Paid softwares, nothing there like only using paid software, you will get good publications. It is not that we're using free software, the reviewer is not going to accept your paper. Nothing like that. All student community, I want to admit this here. I am from a paid software company and I'm telling you this. I'm a reviewer to ACS, Villi and many other Elsevier and others. I never seen and asked anybody asking why you didn't use paid software when they're using Autodoc. But let me tell you what is the main difference. The first normal layman terms difference is paid software, someone is sitting on opposite to you to answer any questions that you raise to them. A support team will be there. That's why you're paying. The first payment is because of that. Someone is there to answer your question dedicatedly one-to-one. When you come for a free software, not paid software, you have to depend on the community, a forum or your friends or some tutorials which is being uploaded by any other people. So that is the main difference. The second difference, as many of you mentioned, features as well as learning curve. Paid software is more user friendly. Click a button and you get that. Single click, it does everything automatically. And in the unpaid or free, you have to do one by one manually. But let me tell you, when I have intern students every year coming here. So I make them learn both open source as well as paid. But the first one they have to learn is open source. And one of the participants clearly mentioned that. Because when you do open source, you really learn each and every step, what is happening in the behind. And once I finish that open source training and then they get into the paid software, they'll ask, so why don't you give this in the first? This is so easy for us. But then I asked them the question, you wouldn't have understood what is happening in the behind when you click a button. So you shouldn't ever think of easy usage. Don't think that I want the scientific software to be easy. No. You want the scientific softwares to be accurate, industrial standards, and it should make science of relevance. These are the three important things which you require. It's not about which is easy. I will use a software which is easy. Okay, what about the accuracy? Have you checked it? Many others are using this software, so I will also use this software. Fine. Are they using the same target protein you're using? Yesterday, I heard many questions on metal ions. Many of the tutorials are not showing metal ions tutorial. Why didn't they show that? And Dr. Hassan clearly mentioned that parameterization of metals a bit issue. So 3D coordinates of the metal ions a bit issue. So that's why the coordination will be difficult. That's why certain metals we cannot do docking. In many softwares, every software. Some softwares cannot handle water. Some can handle water. So that doesn't mean that that is bad, this is bad. It's a very algorithmic challenge. You have to try all possible methods, techniques for your problem, and then evaluate it. Which one is going to give you something optimal? That's what you have to pick up. It's not that a software which is cheaper I will buy. It is not that a software which is being used by hundreds of people I will buy, because your problem is again unique. Your problem statement is unique. So that's what you have to very clearly look at. Now, earlier days, open source softwares and free softwares, the updates are very less. But nowadays, I can see since after the pandemic situation, many people got a lot of time. So they are absolutely updating very tremendously and in par with the paid softwares. Many softwares are getting updated. So, but the new feature implementation, they need funding. Paid softwares, it's like selling drug to the market. When there are needy, they pay for the drug, the pharma gets funding, they invest for the research. The same way for the paid software, when you pay them, they get money from the customers and they reinvest for more features and research. And there are academia companies like Instadoc yesterday, so mentioned, it can come into a commercial software tomorrow, and that money is being feeded for post or paying to postdocs and PhD students. We also do a software called as TurboMold, where it is from the University of Karlsruhe. It is a jump from Germany. Whatever the money they are getting from the selling the software, they're paying the postdocs and research scholars. So that's how it is working on the academia setup. Anyway, keeping all this understanding, let me get into my slides and feel free to disturb or interrupt me in between the session. Let us not wait till the end because sometimes we forget to ask questions. And as Albert Einstein used to say, we are very much curious to ask many things, but we never ask being feeling that is a stupid question, right? In science, nothing is stupid. Some questions will help you to understand and come up with new learning. To be honest, I learn many things from my students and interns because when they ask questions, I sit and refer and learn it, right? Which I might have not thought in that perspective. So always questions helps each others to help and learn things. Moving forward, let us look at a little bit more on the pharma chemistry also, because when you're talking about drugs, it's chemistry, right? It's not biology. But when you talk about medicinal chemistry, it's a combination of biology, pharmacology, chemistry, then pharmacovigilance, quality assurance, everything is coming into the picture. But without biologists, the target identification, validation, molecular level testing is impossible. What will the chemist do just sitting with a compound, right? Biologist only has to take care of it. So you are the people who take care of understanding the problem, which are the disease target like genes or proteins, which is a mechanism that you have to attack, probably activation or deactivation or upregulation or downregulation, and then you attack that specific amino acid using a foreign particle or a foreign molecule called as a drug molecule. It could be a small molecule or it could be a small peptides. And then that's where the role of chemist comes into the picture. But CAD, computer aided drug discovery, could also help you or assist you in that. And yesterday, based on some of the questions, let me tell you this: do not think you can replace wet lab experiments with computer aided drug design or drug discovery. Never ever possible. CAD is going to assist you or give you help to take decisions or guidance to do what kind of studies you have to take forward. But never think that CAD, like ADME toxicity predictions, are accurate. Never they are, but they are going to help you to make decisions. One. Second, don't completely rely on software. Anything, paid or free, whatever it is, and take it in a good sense. Okay, I'm telling you not to discourage you, but telling you the real facts. Give you an example. In calculator and in mathematics, when you say 1 plus 1 is equal to 2, it is always 2. It is not going to change. But in science or in biology or in drug discovery, I have three answers for 1 plus 1. One is 1.9 plus or minus 0.1, 2.0 plus or minus 0.1, 2.1 plus or minus 0.1. All are same for me because I have a error or a standard deviation which I have given as plus or minus 0.1. So unless I have a standard deviation or iterative experiments, you have to replicate it. Multiple iterations of experiments gives you a validation and justification how far the results to be reliable. I'm sure all students, including me, remember during your plus two or pre-degree, you would have done a titration experiment. You would have repeated the three readings to calculate the mean standard deviation. But why are you not doing when you are going to do sophisticated experiments or on molecular level or bioassay level? Of course, it is expensive, right? But uncertainty is a challenge here. So even when you're in experiments, when you're doing only one single run and you're not able to say whether it is accurate or not, while using computers and you're using a word called prediction, how can you be 100% reliable there? Right? So you have to think, you have to brainstorm yourself, justify yourself, whatever I'm predicting is right or wrong. Don't blame it on others. Don't blame it on software. The software predicted wrong, software predicted right. Right? So you have to keep that in mind. Now, one more question. How many of you think X-ray crystallography to get the structures of the proteins is the accurate method? Yes, you can press 1. No, you can press number 2 in the chat. How many of you think X-ray crystallography is the accurate method? 1 is for yes, 2 is for no. If answer is 2, why? Can anybody answer that? Okay, someone said 1 also. It is not the native environment of the protein that is captured by X-ray crystallography. Absolutely right, Gagandeep. Cryo-EM is another solution for that. But yeah, as Pranavatyani said, it depends on the data. Absolutely. So absolutely right. X-ray crystallography is not the most accurate method. And even Vanshika said that those molecules lying in path are predicted but not hidden one. NMR. You're right, Mayank. If it is soluble, NMR is the most accurate method when compared to X-ray. But most of the proteins are not soluble. So that's why we go for X-ray crystallography. See, I don't know how many of you know how when you get a diffraction data from X-ray, how do you actually get the PDB structure? I don't know how many of you know that process. I'll simplify it and explain. When you get the diffraction data, basically it is electron density maps. And then you put it in a software called, that's right, electron density fitting. So, and then you put in a software called as Phoenix or Coot. They're all free softwares. And again, you will do the process of homology modeling. In the normal homology modeling, you're using sequence as your query and a template, and then you model it. But here, instead of the sequence, you use your electron density map, and then you compare it with the existing PDB electron density data, and then you fit it and then predict it. That's why you will have missing residues, missing atoms, and also the resolution also matters there. So don't think that is accurate. It is not the full complete structure of the PDB, but at least the best accurate which we have right now, economically viable. Earlier days, homology models are being submitted to RCSB. Now they don't submit, they don't accept it at all because they know that it just doesn't work. Even X-ray is not good, so how can they go for that? So that is the level of understanding you need to have. We learned X-ray crystallography, we think that is the best. Don't think that. We have to go deeper and understand how that is being done. For marks, qualifications, and everything else, whatever curriculum you are learning is absolutely fine. On real life, you have to read and learn more from experiences and others. It's very, very important. That is where we have to look at the grand challenge for all of us is how to best incorporate the existing knowledge. Everybody looks at what we are trying to everybody looks at what is that existing data we have, and based on that, how to improve it, how to innovate more with a lesser cost. All those things we try to look at. But we also have to look how we have to out of the box, think out of the box. Otherwise, there won't be Nobel laureates, right? When I've been interacting with many Nobel laureates during several occasions we got during my PhD and postdoc, where came to understand that when they proposed this idea to the supervisor, they said impossible. You cannot do this. You're something saying that is out of science, but they made it possible, and that's how they got recognized as a
ಅದು
Nobel.
ಒಂದೇ ಇದ್ರಾಗ ಆಗುತ್ತೆ ಬಿಡ್ರಿ ನಿಮ್ದು ಬ್ಯಾಕ್ಟೀರಿಯಾನೇ ಜಾಸ್ತಿ ಬಂದ್ರೆ.
Oh, sorry. Okay. Now let's have this question also very, very much disturbing to many of them. For example, as a researcher, we think about getting publications, patent, getting a positive result, better activity, less toxicity and all. But a civilian doesn't think about or doesn't care about anything until unless he's very much passionate about research field. The civilian just thinks when he goes to medical shops, understanding, oh my God, what is the cost of this medicine? Is there a way to reduce the cost of this medicine? Apart, this is again a question to you. So apart from reducing the cost of synthesis, apart from patenting, apart from government demands, marketing, increasing the production, do you think, is there a way that you can reduce the cost of medicine within your limits, being a researcher in academia or in industry, with think as yourself, what you can do to reduce the cost of medicines? Any thoughts? You can unmute or you can put it in the chat. So this is the way we have to think to decide which research topic I have to work on. And that is where the entrepreneurship things comes into the picture. Generate crowdsourcing platforms, okay. Finding out cheaper sources. Once a patent is expired, we can, of course, yes, Sandesh, once a patent is expired, we can go for generic, but for the 10 years, you have to pay that high money. May sound different, but can avoid patenting. You're right, Tabassum, to avoid, but how the company will take back the money? Because the pharma industry is spending more than 1.5 billion US dollars for research failures. They have to pay a lot of money for human volunteers. You're testing this this drugs. So where do they get the money? Until they protect it by patent, they cannot reserve that money. That's why they are patenting it. By not with IPR. And someone said finding out cheaper sources. Shika, you have to keep in mind the purity of the compound matters, the activity also. When you go for cheaper sources, the impurities will be more. That's why many of the drugs, generic drugs, are still not much active, which is again produced in India because of the cheaper sources, right? So we have to be very careful on that. Repurposing of existing, recycling expired ones. Swati, the stability of the chemical compounds cannot be recycled to its fullest mode. So it is difficult. Repurposing, again, you have to do research, right? So we can do virtual screening. So testing all molecules available will decrease. Vanshiga, screening helps you to reduce the funnel. That is true. But the problem is at the end, when you get a compound, one or two, when you go for testing, if it doesn't work, then doesn't make sense, right? Maybe we provide certain percentage at lower cost with multiple collaborators or investors. Okay. Maybe in silico modulation, once we get full idea about the mechanism. Okay. So considering all the factors of answers that you have given, thank you everyone. So being very interactive. I know there are around close to 200 people and many are responding. Synthetic biology for small molecule production, scale up the novel biosynthetic pathways. Yes, that's all coming into research. We cannot avoid research, right? But after that research, we have to test it on humans, right? And if human dies, you have to pay hefty amounts for their volunteering. That is what the agreement comes into the picture. But let me tell you a simple word. In order to avoid the cost of medicine, avoid failures. That's it, nothing else. Patenting, they have to do. They cannot survive. Pharmacy industry is not a charitable society. They have to get money in order to do research and get more things on the market, right? So it takes, they need money. Without that, they cannot do. And regarding crowdsourcing and getting more investors, yes, let's say 1,000 investors came to that market. They all need their money back. Why do they invest? They need profit. So ultimately, they need the money, right? So as a researcher from our side, what we can do is try to reduce the failures. When you are suggesting some compounds, ensure that you're suggesting promising and confident compound, not just based on some simple normal analysis. Make some confirmatory test, validate it, and then only say, so these are my 10 compounds which can be taken for further testing. That should be the confidence level. It's not that maybe active, I don't know, unless we test, we don't know. So how can we achieve this kind of confidence? We have to get a lot of possible data with respect to that objective that we are working on. It's not only from software, it's from your science of relevance also. The synergetics between human expertise and computational predictions, including experimental predictions, is going to be really very productive, right? So some more is coming, but let me continue with my slides. Okay. Finding analogs. Yeah, right. Give quality, how we can reduce the cost. Organic sources as a substitute for that drug, which can reduce the side effects probably. Yes, we have to look at also to reduce the toxicity. Government always pitches in, but they have also have limitations because pharma industry cannot go to a loss when they spend a lot of money for research, right? That's where we have to see how the CAD can be evolved and help you in finding this to some extent. I not say completely. So let's look at it. The problem with everyone, including me, we want everyone needs fast result. We don't care whether it is accurate or not. We want fast result. Sir, molecular dynamics, I won't do because it takes 10, 15 days to do the completion of the thing. But you're doing 4 years of PhD, right? Why can't you do the first thing as learning molecular dynamics for the first time? Don't do molecular, don't learn molecular dynamics at the end. So when interns comes here, initial five or to 10 people, when they came, I made a mistake by giving the molecular dynamics at the end. They feel reluctant, didn't do. When the 11th person came, after learning things, the first assignment given was molecular dynamics using some sample data set. So they learn that first and then they move forward. Then they shouldn't say that it doesn't take time, right? So we need to have accurate results than we are looking just for fast results. Of course, fast is also important. And then we need a strong visual focus, which is very, very important. I'll come to that more detail in while progressing with the slides. And we need everything to be easy. Now, I know many students are here. If I ask you, why did you choose microbiology? Why did you choose zoology? Why did you choose botany or biotechnology? Majority of the people will be telling me one single answer, because we found mathematics very difficult. I think many of you agree to me, right? Many of you tell that, sir, it was my passion. I liked it. That's why I took it, right? Now, why did you find mathematics difficult? Because you didn't understand it properly, or the way you took it forward was not in the right way. But when some person understands and makes max easy, that means he or she understood it properly. So don't confuse between black boxing. Black boxing means you click a button, you don't know what is happening in the behind, but it's doing the job for you. It could be anything. Even machine learning is black boxing. AI is black boxing. We don't know what is happening, but it does the job for us. But when you're learning, when you're doing certain mechanisms, make sure that you know each and every step, right? So you think that I don't have time to read manuals. I don't have time to read user guides. I don't have time to read the full paper. Here, how many of you read only the abstract and the conclusion of the paper, not the whole paper? I know many of you are doing that. Even I also did that, right? So we need to have a proper understanding about where to do and what not to do and what to do. So sound science, things of relevance is something very important we have to look at always. Natural consequences, higher pace, algorithmic challenge. As I told you earlier, don't think that single software can or single algorithm can solve each and every problem. It's not possible. You need to have different dependency on algorithms. But in academia, you might think, sir, we don't have that much money to spend money. See, no one in pharma or in pharma industry, just using a single software, they cannot do CAD. They cannot do computer aided drug design. Never, ever possible. If you know someone in the CAD department in pharma, just talk to them. They will say that for docking, we use this. For ADME, we use this. For QSR, we use this. For toxicity, we use this. They have multiple packages. When you come to academia, you might not be having that much of funding to do that. That I agree to it. But ultimately, that's where the algorithmic challenges we have to understand. Non-specialists more involved. That is again, I'll be discussing in my next slides. The chemist will say, I have synthesized some compounds. Can you do docking and tell me what it could be the best one? Okay, what about the target? Do we need to find it? That's a question, right? So the strategy itself is wrong there. The biologist will say, I'm going to work on anti-cancer drugs. I have some plants, I extracted some compounds. I want to make it sure that this is having anti-cancer property. But did the plant, did we prove that it has anti-cancer property? No, again, there is a strategy issue. So that's where the communication challenges are always there involved. So that's where we need to have collaborative and cooperative work. So there should be biologist, chemist, pharmacologist, toxicologist involved in a drug discovery project, even in academic level. Recently, there was a drug discovery hackathon happened by MHRD, Government of India, right? I am one of the coordinator there. So we saw a lot of submissions came in, and that's where we justifiably ask them certain questions that you have to bring in. So you have to give many data that is not simply you can get from a click of a button. You have to have a personal understanding, expertise in that to infer and to conclude what to write there. So that is what you have to communicate with multiple expertise and then come to a conclusion. Basic CAT skills are always helpful throughout the pharmaceutical R&D, but in the right way, not in the wrong way. Now, moving forward, this is a common situation where a chemist sits and synthesizes compounds, looking at good yield and purity, and informatician always tries to do statistical analysis. And when you're going for a modeler, it could be a bioinformatician or a computational chemist, whoever it is. They more look at ADME properties, ligand-based drug design, drug discovery, and structure-based drug discovery. Here, I also want to tell you a big difference here. My master's was on bioinformatics and my bachelor's was on biochemistry. Since I did my master's on bioinformatics, I think I have the privilege to tell this. Bioinformatics is not drug discovery. Keep this in mind. Bioinformatics, mainly we focus on proteomics, genomics, sequence analysis, statistics, ML, computational biology, scripting using Python, Perl, R programming. That is bioinformatics. When you come to drug discovery, it's mostly pharmacology, pharmaceutical chemistry, and a little bit more of biochemistry, right? Towards that. Why I did not include the drug discovery to bioinformatics? There should be a good understanding of chemistry here. very much mandatory. Now, if a bioinformatician want to work on drug discovery, no problem, right? You still can work, but ensure that you have a chemistry friend in that project or a pharmacology friend in your project. In bioinformatics, are you studying ADMET? Yes, you learn what is ADME, but do you learn the mechanism? No. Do you learn what is a catalytic site? Do you learn what is the reaction happening between a ligand and a protein amino acid? No. You learn docking, right? I know you learn docking. You learn docking score, energy. Now, coming to molecular dynamics, it is not even drug discovery. It is not even bioinformatics. It's completely biophysics. You're talking many things in biophysical terms, RMSF, energy terms, time frame, many things. But it can be helpful in drug discovery. So we have to understand the communication challenges here. But I never said that bioinformatician cannot learn drug discovery or he or she cannot learn molecular. You can. That is your passion and interest. But keep in mind, you need to know basic physics and basics chemistry, including mathematics. So when you're going for bioinformatics definition itself, it's a multi or interdisciplinary subject or a field where we are trying to solve biological problems using mathematical and statistical approaches with the help of programming languages. That's how the best way I can define it. So make sure that that's the only way I have changed my track. After bioinformatics MSc, I thought that I'm not a good programmer or a scripter or not going well with my computational biology or genomics. I changed my track because my interest was more into biochemistry, mechanisms, and drug discovery. I changed my track, and my PhD was completely into computational, right? Into chemistry and medicinal chemistry. So we have to make a understanding here, because when you up to MSc, everything is fine. But when you make a decision on PhD, things are going to change. Interdiscipline is always welcoming abroad. In India, slowly it is coming up, but a little bit not so much in the state universities, but Indian central universities and other institutions, they're absolutely welcoming. So there is a common situation gap here. As I told you, a modeler will be trying to design a compound from database or fragment based, and then ask a chemist, can we synthesize this? Chemist will say, oh no, it's very difficult to synthesize, very expensive. Chemists will synthesize some compounds, making derivatives, what is within their, what they can do, and give it to modeler. Modeler say, no, no, this is very toxic. That is what is predicted. Activity is bad. Docking is not going well. It is not entering the pocket. So there's always a communication gap between both of these expertise. Now let's see a better situation where chemist understands what is toxicity. Not in deep, but at least visually. Chemist understands, okay, docking has happened. Now this region, I have some space left. I think I can make a derivative there so that I'm sure this is going to fit there. Or chemist also have, the modeler helps the chemist to understand this region of the compound is being very toxic. So can you think of optimizing that region so that don't touch any other, only try to make a functional group or derivative on that region. That should be the focus. And modeler also understand from retrosynthetic pathway prediction what a chemist can really synthesize and what she or she cannot synthesize. That is also important. Otherwise, modeler will be simply predicting thousands of molecules with many things, and then ultimately chemists will say, no, I cannot synthesize. The other perspective here, most of the biologists will do, we will take a database which is purchasable. We do all the predictions and we do completely purchase a compound and test it. Now, the question here is whether you want to publish or whether you want to make money. Publishing can be done with all these purchasable compounds and getting the activity predicted, and you have a better than the references. If you want to make money, you have to patent it. When you're patenting, you need at least for a better patent, for making money, you need at least a novel molecule there. There, chemist comes into the picture, right? So for patent means money, not to put in your CV, right? And publications is actually your transfer of knowledge, and you're getting to know about your expertise, and other people get to know many things about you. right? So that's where Thomas Kruse Lipinski, I know everybody have heard about Thomas Kruse Lipinski, that is the Lipinski rule of 5. So he has said a rational dialogue between chemists and the modelers will be very, very productive, right? And he said this in 2004, almost 27 years back. Oh no, 17 years back, right? So this is what being said earlier, but are we doing now? We are still trying to do one-man job, or we try to collaborate within our vicinity, but we have to tremendously form a consortium and collaborate. It's not that when you have a PhD student, no need to wait for your professor to collaborate. You have to proactively find out the people and then check with your professor, can we collaborate? That is a proactiveness, right? You're a PhD student, you have to find your roots for your research. Now, coming to the next point is that what kind of approach we can apply to drug design. This completely depends upon the data availability. If you don't have the right data, then don't do it. Now, what is the title of today's topic of my presentation? To find the bioactive conformation. Why did I mention bioactive conformation? When you have a ligand or a compound that is being synthesized, it has a structure. If it is crystallized, you might get the crystal structure, that is the 3D form. If it is not crystallizable also, you will be able to find the probable structure geometry. Now, whatever that synthesized and stable structure that is there, but when let's imagine that is safe to consume and you consumed it, administered, and when it binds to a biological target, the conformation of that ligand will change. So the conformation that binds to the protein or the target, that is what we call it as bioactive conformation, right? So the conformation when the compound as it is and the conformation when it binds to a protein, both are different and it cannot be seen. While doing molecular docking, we are trying to find out this bioactive, the right, perfect, and comfortable or stable bioactive conformation within the protein. That is our goal for doing molecular docking. Now, proteins, you need to have 3D structure of proteins, which is from extracrystal structure. Homology modeling, you could have found many papers published using homology modeling and then docking. But let me tell you, many papers, I wanted to ask them this question. Did you check whether the key amino acids in your homology model protein, whether they have the exact identity in the template, whether there is any missing residues in that region? Is there any missing atoms? What is the quality of that particular structure, especially on the binding side? Because they never mentioned about all these things on the homology model protein. And many of them did not even do at least 20 nanoseconds of molecular dynamics on the backbone of the homology model structure. So when you are not knowing the stability of the particular model structure, and then you're going and doing docking, so already your homology model has an error. And top of that error, you are again doing a prediction, you're doing molecular docking. So you are increasing the level of error. I don't say this is wrong, but try to do whatever you have to do. Right. So when you have 3D structure of proteins from X-ray crystallography and the ligands, that's where you perform structure-based drug design. And let me tell you, just doing molecular docking doesn't make sense because your protein is always rigid here. In real life, your proteins are not rigid. They're very much dynamic. So you have to also confirm that whether your ligand can be stable even though your protein is dynamic. If your ligand can still be within the pocket at least minimum 100 nanoseconds, then your docking makes sense. And the final energy that you get out from molecular dynamics is your actual binding energy. Whatever other binding energy you are predicting using Autodoc Vena, CSAR, or any other softwares, they are not actual values. They are not, because I said they are not actual value because your protein is rigid there, right? So molecular dynamics is a non-avoidable mandatory step after molecular docking. And if you're not doing that, no. I can recently see there is a lot of paper published in JBSD. right? I don't say the journal is bad, it's good, but recently I saw that due to lot of self citation, they removed the impact factor. So try to avoid that. Even if reviewers are asking to do, you have to cite this, don't cite it. Cite a relevant other paper. Why you should get into those track of self citation? Self citations are not entertained. That doesn't count your H index. That doesn't count into your I index, I10 index. So keep in mind, avoid self citations. Very, very important, right? Anyway, so that is what structure based drug design. Let's imagine that you have 3D structure of protein and you explode all possible ligands and nothing is docking. And then you have to do a fragment based approach, right? But when you're doing this, please, this is mandatory. I'm telling you on reality. Don't think that why this guy is saying this, I wanted to do it. But tomorrow, if you want to patent or if you want to do a good quality research, my question is whether you want to publish 100 papers or whether you want to publish just 2 quality papers and sit aram say get a better job. I know a friend who just published a single paper in Nature, 1 patent. That's all he have. For the past 14 years, he has not published anything else. He's in a very top position in a company. That's it. That paper got him a job. Do you want to do that, or do you want to publish 300 papers and still live a life with our research? So that we have to take a decision. Why I want to say that when you ever do fragment based drug designing or scaffold hoping, make sure that a chemist is sitting next to you, because fragment based drug design, everything looks very nice on computers. But if you want to do the testing, bioassay, animal study, in vitro or molecular level, you have to make this compound, right? So computers, to some extent, they know this is makeable or this can be synthesized. But only a chemist can really tell you whether he can synthesize it or not. So when a biologist do scaffold hoping de novo synthesis, ensure that you recheck this with an organic chemist who knows to synthesize. They let them confirm that this can be synthesized or not. Otherwise, don't ever do fragment based drug design just based on computational predictions. Right?
Dr. Giri,
Today, yes.
is it absolutely necessary to do MDS? Even if we are going for wet lab validations for our compounds, say we have around 10 lead compounds, we want to go for wet lab validation. So is it still necessary to do molecular dynamic simulations?
Very much important, madam. Very much important.
Why?
Because, as I told you clearly, because in your protein, you made it rigid in docking. Any docking program, your protein is always rigid. Your backbone is not flexible there. You made it very static and rigid. But in a real life, protein is not like that. It is very much dynamic. So you have to and also when you're doing docking, while you're performing docking, I'm not talking about the binding energy calculation. While you're doing the docking, you made it vacuum. There is no solvation there. So your protein is not in the solvated environment, even though there's an imaginary environment, but it is not actually the solvated environment. But your protein is actually in a solvated environment. But molecular dynamics takes care of solvation, and it makes the protein flexible, dynamic. It also applies pressure and a temperature to look at the equilibration. This nothing of them is applied in docking.
Yeah,
so
but
there are
but if wet lab validation you are doing, then all those things are taken care of.
Again, I have an answer for that in my coming slides. So when you say molecular docking and wet lab studies, you're finding only a hit molecule. Means you're already imagining that your compound reached your target and it is docking. That's it. But the problem here is your compound has to reach the target, right? So when you're consuming orally, it has to pass through several metabolic pathways, absorption rate, permeability. After that, it has to reach your target. And if it is reaching your target, and if you're not testing them dynamically, and if it is going to break down, then doesn't make any sense. All your hit molecule never convert to a lead molecule. So when you make it dynamic, there are many case studies where they have said, docking, you got the topmost priority compound, but when it came to dynamics, they are getting out from the pocket. They are not staying within the pocket. So after 20, 30 nanoseconds, they're going out. In some cases, 20 nanoseconds, they slightly go out and they go back and they stay there. That is fine. But when they go out and not going back, that is a problem. So this
Okay.
you
So
can only understand from dynamics.
how long should we perform the MDS then in that case for the dog
So
poses?
for a protein ligand interaction, minimum is 100 nanoseconds, minimum.
But it's very difficult to do for large number of molecules.
I absolutely agree with you. That's the reason I have a case study to show you. It's not to do for everything, only for maybe the top 1% or top 2%, which is like 5 to 10 molecules. Now, earlier days, computational, we were having a lot of challenges. But nowadays, I'm sure when you're having funding, the first thing you have to set up is your computers, at least with a GPU card and all those things. If you have a funding of 1.5 lakhs, I myself build a computer with 1.5 lakhs with GPU cards, where I can run 50 nanoseconds per day.
Per day?
So, per day.
Okay, so that's
So,
good enough. So what is your GPU, like 10 GB or how much?
So GPU is not about how much memory. So I have only 6 GB memory, but it is the card with a number of CUDA cores. So RTX 2080 is what I'm having the card. The number is RTX 2080, but the latest one is RTX 3080 is there. Even I have RTX 2060 also. So they have a 3000 CUDA cores. That is going to help you to scale up it. And at least a 16 core machine, 16 core CPU machine. But now now students might think, sir, I don't have this much money. I have a laptop with i5. Yeah, for those people, I'll be showing you some resources at the end of this slide, how to learn molecular dynamics even on your mobile. On your mobile, you can run a molecular dynamics with 129 nanoseconds per day free of cost. But there is a catch, only for learning purpose, not for your research. I'll tell you why and all when I talk about it. But ultimately, ensure that at least you collaborate or pay for someone for doing molecular dynamics. That will improve your justification of your molecular docking.
And after doing MDS, still you will have to validate.
Of course, yes, absolutely. But after doing MDS, you will get almost exactly the trend will be maintained. Whatever coming out of from MD simulations, the same trend you will follow in your wet lab studies also.
Okay.
But the docking will not follow the same trend, most of the cases.
I'll get back to you on the configuration for MDS simulations.
Absolutely.
For the workstation. Yeah.
Sure.
Thank you. Thanks a lot. One more question is there.
Yes.
And there is a question which is there in the chat box, which is relevant here, I guess. It will be very nice of you if you can answer it here.
I'll go through it.
When we use Autodoc, we just upload the protein and ligand, but isn't it possible that protein has more than one docking site, and the site at which the ligand is binding is not?
You're right. For reference, we say in case of enzyme substrate active site and allosteric site, is it possible that the ligand I'm studying is binding to allosteric site or not? Absolutely right, Muskan. I will be discussing that in my next slides. So you really touch based it.
And Mohammed has to say that Schrodinger provides solvation environment.
Mohammed, I clearly emphasized it. While doing docking, no solvation. While doing binding energy calculation, affinity calculation, solvation is there. That is the difference between clicking a button and you're doing manually one by one. So in Maestro, you have a workflow. You click a button, it takes care of everything. Your XP docking, fine docking, protein preparation. So I said specifically when it is carrying out the search points for docking, not solvated. After that, it gets solvated to calculate binding energy. Same with CSAR also. When it tries to binding affinity, it will solvate it. So I told you specifically. Yeah.
Just one more question. So for X-ray crystallography, when we were discussing, you said that for soluble protein, NMR is done, and crystallography, can it be done for the insoluble proteins?
Yes.
But if the protein is insoluble, how will you get the native conformation?
See, until unless the protein gets crystallized, you cannot do crystallization, right? X-ray crystallography. Yeah. But I think I have to recheck on that. I will let you know today afternoon when during the
Sure,
workshop.
sure.
Yeah, I have to recheck that.
Thanks a lot.
No problem. So going for the next, let's imagine that you don't have a 3D structure of the protein, but you have activity data, or you have the bioactive conformation already known from the drug bank, and or you have some other data where there is no protein. In that case, you will do pharmacophore. When you're doing pharmacophore, ensure that your reference molecule is a co-crystal or a crystal data, bioactive crystal data. If you don't have any of them, try to take the best bioactive conformation from the docking data. That is mandatory. Otherwise, your reference does not have any meaning while you're doing pharmacophore. And when you're doing 3D similarity also, the field topology analysis, there also you need the right references. And when you come to QSAR, one of the most dangerous method in cheminformatics and drug discovery. It sounds easy for you, but when you take 100 papers, only 4 to 6 of them. Other papers, not reproducible. I didn't say, many experts and reviews used to say, right? So when you do 2D QSR, it's very, very straightforward. Nothing bias situation is there. But when you do 3D QSR, my question here is, what is that 3D? In most of the cases, I know people take 2D database, or they draw the structures and then convert 2D to 3D, and then they use MOPAC, QSite, MPAC, MMF, or PADL, many other tools to generate the descriptors, right? All are good. Now the question is the 3D geometry. When you're doing QSAR, it is quantitative structural activity relation, structure activity relation. So you want an active conformation. You want a bioactive conformation. We don't want simply 2D to 3D conversion. In most of the papers I have seen, they are doing 2D to 3D conversion. Even I have also published earlier days with 2D to 3D conversion. Later realized that doesn't make sense. You have to take the bioactive conformation. Let's say you don't have it. Do docking, extensive real docking, and then take those poses as your confirmation to calculate the descriptors. So in my final paper before my PhD, I have done that after realizing that is a mistake. So when you have sophisticated software, don't think that everything can be done click of a button. When they're teaching the tutorial, they're clearly mentioning this. But we, most of the time, we remove the scientific part. We just look at the pictures and recreate ourselves on the tutorial. So students, be very careful, make sense when you when some instructions are given why you are doing what you're doing. So 3D geometries which you're using for descriptor calculation, especially for biological activity, take the bioactive conformation. Let's imagine you don't have a protein, right? You don't have to dock, right? You cannot dock. In that case, take a reference molecule which has a bioactive conformation. Try to generate conformation analysis where it can match similar to your QSR, 3D conformation, where you can do pharmacophore mapping also can be done, and then generate the descriptors. Either way you have to do. Otherwise, the 3D that you're generating, the 3D descriptors that you're generating from as normal 2D to 3D doesn't make any sense. Because for chemical or physical 3D property, that makes sense. But for biological, I told you already, when it binds to a target, the bioactive conformation matters here. So in that case, we call them it as ligand-based drug design. Let's imagine you don't have both protein and ligand. You tried everything, but now you have to do a high throughput screening. It's not simply taking a protein and taking multiple ligands. It's called high throughput screening. High throughput screening means multiple targets, multiple ligands, and you do an ensemble. So that is what high throughput screening, which is called CombiChem, combinatorial chemistry. Now to answer Muskan's question, I will do it here. Okay. When you want to explain what is molecular docking, my professor used to tell me before my defense, and also my guide and supervisor who was there in University of Kerala used to tell me that, Giri, if you can explain your thesis or your research to a grandmother who does not know about research, then you're good to explain this to anybody, right? That's where we have to look at how we can quantify or what we can understand from molecular docking. So molecular docking is basically to determine the optimal binding structure of a ligand. So optimal binding structure means I am trying to find out the bioactive conformation to a receptor. It could be a target, a protein, or DNA. And then we try to quantify the strength of the ligand receptor interaction. That is what we are trying to do. I'm not trying to find out the score. I'm not trying to find out the energy. This is all for helping the quantifying it. That's all. But this picture, it's a very good picture that I have taken from enzyme methodology book, which is really, really important for those who are writing papers and explaining about molecular docking. Let's see what is we are trying to find out from molecular docking. We are trying to find out where the ligand will bind. As Muskan rightly asked, there might be multiple pockets in the protein. Where will the ligand will bind? How will it bind? It can bind in several ways, but how will it bind? How strong it could bind, and why it is binding, and what makes a ligand bind to the receptor better than the others. Because if you have a very big pocket and you have all small molecules, everything will go, everyone will go and bind. Now you are in a bias situation, which one I have to choose? So what makes it very specific, this ligand is better than the others? And like that, many other questions mechanistically looked at. So when you say drug discovery, you have to look at a mechanistic driven drug discovery. Search for this word, mechanistic driven drug discovery. Very important. Now, let me go through this picture so that I will explain each of these questions. First thing, catalytic function. You need to understand what is the specific region of your protein is having functional site, active site, motifs, domain, or any catalytic site. Very important. It's not that you load a protein, you use a binding pocket finder or some other tools and then dock to the first pocket. No, that's why I said biochemistry is also important here. You have to understand the mechanism. Here, when the students used to come, they spent almost 3 weeks to understand the mechanism to choose the right PDB. But they're very urged to choose the PDB and start doing docking. No, you have to understand the mechanism. Second, regulatory mechanism if it is enzyme. Are you targeting active site or allosteric site? So until unless you understand the mechanism, how can you decide that whether you're going for an active site or allosteric site? What I'm trying to say here, the mechanism is when you take a pathway, right, a biological or a metabolic pathway, you have receptors. You need to understand what is the role of this particular receptor in your disease or for particular activation or upregulation or downregulation. You have to understand. And then you have to also identify which are those amino acids in that particular protein target or that receptor, which is a key factor to downregulate or upregulate the mechanism. And that is the amino acid you're going to target. So you have to understand the catalytic function first, then the regulatory mechanism. The third one is cleft detection. That is what you're doing, pocket finding. There will be multiple pockets in your protein. Doesn't mean that the largest pocket is your binding site. Not necessarily. Sometimes yes, not necessarily. There will be multiple pockets. Now, which is the right pocket for you? The answer is from the first two questions, catalytic function, regulatory mechanism. When you understand which is the key amino acid for my functional site or for that particular mechanism, if my pocket 1, 2, 3, 4, which of my pocket in the protein is containing these amino acids, that is my binding site for my project. For other project, the same protein, they might be focusing on a different pocket because that is the different amino acids for a different mechanism. A single protein can have multiple mechanisms. There are cases. So that's how you identify and choose the pocket. It is not that avoiding and overriding catalytic function and understanding without understanding mechanisms, you directly go predict a pocket and do docking. Doesn't make sense. So as yesterday Dr. Hassan mentioned, you have to go through papers, literatures, understand, drill through all the details of the protein, understand the mechanism, a lot of reading involved. That is the review of literature that you have to do. Not finding that, sir, many of the people have used this PDB for this disease. I will also use the PDB for this disease. Do you want to repeat the same mistake others have done? We don't know if it is mistake or right one. With my PhD student, one paper, we clearly mentioned this. These many papers mention this, but the mechanism is wrong there. And we have many other supporting documents to say that that mechanism is wrong. That means that choice of PDB is wrong. So that's the way you have to come up, right? It is not to harshly tell people that this is wrong rather than telling this is the right, positively. So ensure that you understand a protein or a receptor's catalytic function, regulatory mechanism, and then choose which is the right pocket for you. Not necessarily the first one, it could be second or third. Then geometrical analysis, whether there is any conformational change when your ligand goes and binds it, can be only studied through molecular dynamics, not through molecular docking, because molecular docking, your protein is stable. Now you might say, sir, this software has flexible docking. That's all. Yes, they have flexible docking, but not dynamic state. It is all MMP BSA methods. They are all not as accurate as molecular dynamics, right? But saves time, absolutely. Then flexibility. When you're coming to flexibility, you have to look at whether we are targeting the aposite or substrate bound set, or we are attacking the substrate bound set so that substrate doesn't go and bound. Rather, we are inhibiting the binding of the substrate with a molecule. So that also we have to understand the mechanism. Then we are looking at conservation studies. Conservation studies is most of the time we never look at. For example, anti-cancer drug, you do MTT assay. In many papers I have seen, no one is worried about checking the MTT assay on healthy cells. They always target the anti-cancerous cancerous cell line and see whether there is activity or inhibition on the particular compound. Now we have to also make sure that this does not help disturb our healthy cells also. So where is that? That is called conservation, selectivity. While you're doing docking also, you have to do the same thing. So for example, DPT program by NCI US, National Cancer Institute. You can send yearly two compounds to them free of cost. You have to pay only the shipping charges. You have to apply for a funding like that proposal. If they agree, yes, you can. And then they will test your two compounds on 96 cell lines, 96 cell lines. Basically 60 only, if required only they go for 90, but by default it is 60 cell lines. They test on all cell lines. Unfortunately, the data will be public. So if you're planning to patent, you cannot, but otherwise for publications, you can. It takes time, but yeah, both. That tells you the selectivity, specificity, and sensitivity. That tells you to understand how selective your compound to that particular cell line. Right. So, and also one another thing is that when it docks with many other proteins, you will say my compound is multi-targeted. Come on, how can you say that? Just simply docking just says multi-targeted. We want to analyze the pocket variances. So that is called ensemble search. So we want to see the docked protein pocket, how far there are other proteins which is having the similar binding pocket. They are all the proteins where your ligand can go and dock. In CSAR, you have this functionality. You can go and dock. So they're all the related proteins. They're also your ligand can go and dock. That is why I told you also indirectly, but let me make this statement here. In early 1990s and all, your docking always we explain it as key lock hypothesis. It is a key lock hypothesis. But in 2021, no. Single key can fit into multiple locks, which I told you the example right now. Similar binding site can be there in multiple proteins. It can dock there also. And a single lock can have multiple keys also. If a lock is a bit larger, multiple ligands can go and bind. I mean, one by one, but there are more chances of multiple keys can fit in. So it is only halfway the truth lock and key hypothesis in 2021. So that is what you have to have a better understanding. As Purvi correctly mentioned, we need to have prior knowledge of the sequence present in the active site and make a grid around it for binding. Absolutely, Purvi. So there is a tool called as ProSite, online tool. ProSite, you can paste the FASTA sequence of your PDB, and it will try to search motifs, domains, functional sites, active sites, searching the protein family databases, and then it will try to identify which are the signatures. So it will also tell you which are the region of the amino acids, which is key amino acids for this particular searching. So ProSite is going to help you with the knowledge of the sequence. You will be able to find that out. Okay, so that is about the conservation studies. Then surface characteristics, whether your surface related to the pocket and near to the pocket is more hydrophobic or lipophilic or solvent accessible area, polar surfaces area, all those things has to be understood. And then finally, we talk about interaction energy. Now, when you're talking about interaction energy, that's where the solvation comes into the picture. So when you're talking about an energy, it is delta delta G. Why I said 2 delta? Because it's a difference between 2 delta G, right? 1 delta G is taken from solvated state. Another delta G is taken from desolvated state, both required. So when you click a button, you might not understand, but software does it automatically. So one delta G is from solvated, another delta G is from desolvated state. And that is why I said delta G is what you're getting for your binding energy. We'll come to that bit later. So the challenges here is we have only 2 questions to answer if you're looking only at docking. I'm not talking about affinity energy. I'm just talking about docking. What is the pose of the ligand in the pocket? That's all the first question. Second question, how happy is your ligand there to score it? Now, how many of you think that docking score is very important to choose the final 2 compounds? For yes, you can press 1. For 2, no. How many of you think docking score is important to select the final 5 compounds? Again, I'm repeating, final 5 compounds. Okay, many of you said yes. But the answer is no. Right? The answer is 2. To choose the final 5 compounds, docking score will not help. Let me come to that again. I know I am confusing you. Let me justify it. First thing what I want to mention here is that when you are doing virtual screening, you will have thousands of molecules or hundreds of molecules or millions of molecules. So when you have millions of molecules or thousands or hundred, immediately if you want to select top 10% of the molecule or 25% of the molecule, the best way to sort and screen is you just take the best docked score molecules. So you click a button to sort, and the top 25% or top 10% or top 25, whatever, you will be choosing those compounds with the highest docking score will be taken. That is what the first filtering you have done. After that, again, you will redock, right? After redocking to check whether the scores are correct or not, or they're just reiterated. And after redocking, you have to go through one by one to understand whether these structures are interacting with the key amino acids based upon the mechanism as well as the catalytic site. Then you will filter a few of them. Some might interact, some might not interact. You filter a few of them. And finally, you end up might be 60 or 50 compounds. And for that 50 or 60 compounds, you have to do a rough MMPBSA kind of study to understand the binding affinity and the binding energy. Because this 50 or 60 compounds, you're very sure because all these compounds are interacting with those key amino acids, which is based upon the mechanism. They're also having optimal docking score. And finally, 50 to 60 compounds, you are also again doing MMPBSA. It doesn't take much time, maybe less than 1 hour. It will take, not a molecular dynamics I'm talking about. After that, it will try to give the values of binding energy as well as the affinity range. Then you will choose the best 5 or 10 molecules, not one molecule, 5 or 10 molecules. Now, your best 5 or 10 is completely dependent upon the energy and the affinity range, not the score. The docking score has more importance when you're filtering compounds after virtual screening. That's where you have something called as enrichment factor. So in drug discovery hackathon also we have asked for enrichment factor. Many have submitted the data. Many have asked them what is enrichment factor. They learned it and then they calculated it and they submitted it. So an assignment to you, everybody, to students today afternoon, tell me what is enrichment factor. So when you're doing virtual screening, it's a question, how many of you report enrichment factor in your paper? That tells you how far your virtual screening is effective, right? So that is very, very important when you're doing. So docking score is mainly for virtual screening, but not for selecting the final 5 or 10 compounds. Keep that in mind. And it is not that one single docking is going to get you the data out. You have to do iterative, repeated docking. And initially you have to confirm the parameters that you're given for docking is also correct, because certain parameters will change. Let's say autodoc, the American algorithm. How many of you have checked the parameters? There is clustering parameters, confirmation parameters. In genetic algorithm, they have runs, population generation, iterations. How many of you tried changing and played around and understood what is the difference there? So don't think that always the default value is going to work for all the proteins. For learning purpose, absolutely fine. I will never accept a paper if someone is sending for review anything less than 1,000 or 10,000 of runs in genetic algorithm for autodoc Vina, or at least more than 10 runs is required. Otherwise, they will not explore the clustering criteria because there will be a lot of clashes, intra clashes, and so in order to avoid that, we need many runs, right? So this is very important that you have to understand the strategy. Now, another one more question. How many of you apply Lipinski rule of 5? Let's say you are taking database, and how many of you apply Lipinski rule of 5 in the initial step itself to trim out compounds? So for yes, you say 1 and no 2. How many of you apply Lipinski rule of 5 before doing docking to for filtering the compounds? Okay, fine. And how many of you have read Lipinski rule of 5's paper? If yes, 1 and 2 for no. Lipinski rule of 5 paper, the actual paper, how many of you have read? Okay, many of you have not read it, right? When you're purchasing a smartphone, you go through the website 100 times, you speak to people, you speak to them for the features. But when you're doing the research, why are you not reading the paper? I'll tell you, this is a serious issue. In earlier 1990s and all, before 2000, the oral drug failure was mostly because of ADME and efficacy. But later, when you have all these sophisticated tools and many things were being automated, later 2000, the oral drug failures was mainly because of strategy, means unnecessary usage and usage of certain rules and methods for a non-desired steps. I can see only 1 or 2 people have read Lipinski rule of 5 paper, others mostly not. Then why are you using it? Now you'll answer me, sir, most of them used, so I also repeated the same thing. Why you want to repeat the same mistake others have done, right? So you have to, before using something, justify yourself whether this is required for my work or not. That is very important. I'll tell you about these problems in the coming slides, right? Anyway, great. Thank you so much for being very responsive. So let us go through the steps involved in the CSAR package, especially when you're looking at docking perspective. The first step is protein preparation, right? What all things to be looked at protein? It is a X-ray crystal structure protein. Do you need to minimize it? Question. You most of the time, yes, sir. If I ask you why, because the software I'm using, they asked to minimize it. Okay. Now, my question is, when you put your force field for minimization, force field creates artifacts. Do you want force field based minimization, or already a electron density map based crystal structure is already available? Why you want to disturb that? Minimization is actually required in a protein structure only if you are modeling the missing residues or missing atoms. So you will do a modeling again. For those modeled residues or atoms, you have to do minimization. That is called restrictive or geometry restraint minimization. Basically, you're freezing the other part and the rest of the part you're only minimizing. You need not to minimize the whole protein, not required. Why you need to disturb the whole extracrystal structure? The second thing is that you have to add hydrogens. That is called protonation. Now, adding hydrogen is easy job, right? Just simply add, it will add. How many of you thought about the bond angle? How many of you thought about the direction of those hydrogens? Bond angle anyway will be standard, right? Between oxygen, carbon, oxygen, hydrogen, or carbon, carbon, hydrogen. Those bond angles will be standard. That will not go beyond anything else. But the problem is direction, the orientation, where your hydrogen atom is facing. How many of you thought about it when you're adding hydrogens using a click of a button? Because this direction of the protein, direction of the hydrogen is very, very important, right? So, Prakashja, I will come to that question for the ADMET calculation, why it fails and all. Yeah, I'll come to that. So, and also please remind me during the place where I'm talking about ADME. So, the direction matters a lot because the hydrogens are not there in your crystal structure. You're adding it. If the directions are not coming properly, your predictions are also wrong because your interaction might not happen. A slight flip on the hydrogen atom is not going to generate an interaction there. So how many of you thought about it? Because I click a button, it gets added. I don't care what is the direction. So this is the level of depth you have to go for all these wet lab studies, dry lab studies. Now you think, sir, why are you making it so complex? Yes, drug discovery is a complex. If you think it should be easy, do the way you want. But tomorrow, when you wanted to go for a production level, then you will think, yeah, I have heard this somewhere that we should have applied this. I should have learned it earlier. To be honest with you, many of the things I learned after my PhD, not during my PhD, because we didn't realize that. I'm talking something almost 9 years back. We are realizing when we are applying it to certain real life experiments. So there is an opportunity you are getting learned. Yeah.
Dr. Giri, will the software will not take care of the proper energy minimized state of hydrogens when it is adding the direction?
The question is, are you aware that the software is taking care or not? That is a question. Not all softwares are taking care. For example, MGL tools, they also do add hydrogens. But the directions always you can modify.
Okay.
So
and then how do we know that which one is the best direction?
So when you're looking at your co-crystal ligand, when you have a co-crystal ligand only, I can comment this. When you have a co-crystal ligand, that's where the direction that you can customize it. And there are some real good papers where they have talked about the flipping of atoms, where they have told about the difference between the flipping of atoms and unflipped atoms. I'm not talking about amino acids, I'm talking about atoms. And there you see the difference in the binding energy also.
Okay.
So yeah.
I had about the geometrical analysis, which you discussed earlier.
Correct.
So it is like if you have a co-crystallized structure, and you analyze it on MD simulation before you do docking, without ligand and with ligand.
So not before docking, after.
Which
docking.
one you should take?
After docking.
So what you should take for your docking? What you should take for your docking is just simply the structure that is available, co-crystallized, which
no,
has.
madam. Let me explain. This is not applicable for all the studies. For example, recently when we looked at the COVID structures also, we have a open state and a closed state conformations were there.
Yes.
So so that is where it is matters. So there they solved the structures properly, so we were able to understand the closed state and open state. But when those structures are not solved, you have to understand whether there is a difference on closed state and open state on the conformations. And that is not sometimes necessary for every docking studies. Depends upon the protein properties. So for example, if it is more flexible and you have a position where there are closed state possible, then you have to look at it. So when you read the PDB papers, you will be able to understand whether they have reported it or not.
Okay.
Not required for all the proteins.
Okay, for those proteins where open and closed conformations are not there, you can just simply take a structure and preferably the one which has a co-crystallized ligand.
Yes.
And
then you can do
your
the MD
docking.
simulations for open and closed.
Okay, thanks.
And it is a multi-step. It's not a single step. So you have to manually create the state and then minimize and see which one is stable. So it's
For
not
the
a
protein,
straightforward. Yes,
again, for the proteins which have open and closed conformations. For example, spike protein in SARS, if you're talking about SARS.
Yes.
Okay.
It takes time, a lot of resources required. So depends on what you want to look at.
Sure. Thanks.
Now, initial step is we use protos for protonation with hydrogen, and also we take care of tautomerization. And also we wanted to select the water molecules which is there in the protein. There is one more question. Let me check it out. Okay.
And mommy urcha. Yeah.
Okay. Protein structure of X-ray crystallography of protein, most of the time atoms are missing. So how we know which and how many atoms are missing in the binding sites? That's where you have to do. Yesterday, Professor Dr. Hassan was mentioning about DALI, and there is one more called VASTA. And then also you can check protein descriptor calculator. I forgot the name, where they tell you is there any missing atoms or all those things. So that will be able to tell you if there are missing atoms on your binding site, not binding site, missing atoms overall, and missing residues also. So most of the time missing residues will be reported in the paper of the PDB. Yesterday, Dr. Hassan showed you how to go to the DOI and all. So you can look at the paper to understand whether they have already reported about the missing residues and all. If there is missing residues, you have to model it. And after modeling, you have to minimize that region, what you have modeled it, and then you go for docking and defining the structures and other things. Okay. So here after the water selection, you have to do optimization and score, and then final selection is completely based upon visualization, which I will show you, and then you can do optimization. Optimization is again for chemists, or you can do fragment-based drug designing, which is based upon torsion analysis, explorability computations, and high visual affinities. Now, what it tries to do for ligand as well as for protein preparation, it checks with the tautomerism, whether we also do not want to use much of homology modeling, because it's usually worse if you have not done all the pre-checks. And a co-crystallized molecule is always good to start, but if you don't have, then you have to manually define your binding site based upon the amino acid as well as the pocket size. Then pseudo rotations, especially in histidine. This is what I was telling about the directions, wrong element assignments when isoelectric. These all matters a lot when you look at your binding pocket. And water selection, tautomers, hydrogens automatically being proposed. That's what I said. Software takes care of it automatically, but you have to also make sure they taking care of everything. So the hydrogen network for every pose, whether it is affordable or not, it will concentrate on the binding pocket and try to understand if there is a co-crystal ligand. A real interactivity is also possible, which I will be showing you in a moment. Now, what it really happens when you click a button of to dock? All these green colored are the amino acids in your binding pocket. The white color is your complete ligand. In earlier days, there's one more step involved. You have to generate conformations and you have to use that conformations to do docking. But as the technologies and algorithms improved, we were able to generate conformations within the pocket itself. For that, here we use a fragment methodology. What we do is, based upon the linking and breaking rules in organic chemistry, we try to break the molecules to make fragments. So within the pocket, it will try to place a first fragment in different locations based upon the interacting points. When you define the binding pocket itself, it will identify the interacting points. And based on that, it will generate all possible conformations, right? And then one by one, the fragments will get attached. So this way, you will try to avoid intra-ligand and interligand clashes. You will also avoid overlapping of your molecule to the amino acid pocket size, and it will be much faster also. So this is the algorithm that is being used within CSAR in order to do molecular docking screening. So it will generate as many poses, right? That is what it is being done here. Now, how to calculate binding affinity and binding energy? That is also another question. Okay, so how do you minimize, affinity and binding energy? As I told you earlier, we are taking delta G. First, the delta G, free binding energy or Gibbs energy of unbound, the ligand unbound in water and ligand that is bound in the complex with water. And we take the difference. Difference means usually addition when it comes with minus, negative, negative. And then we have the, if you have the highest negative value in delta G, that's what we call that ligand is much more stable there, right? And now we can always see the desolvation. I told you, solvated state and desolvated state. Your ligand might be very unhappy when it is taken out of water. And also it might be still very unhappy to not to bind with water anymore. So sometimes your ligand is not happy with water and without water, both situations. In some cases, it might be very unhappy to get locked into a pocket, but it might be very happy to be freed out of the pocket, both situations. So all these ensemble or these situations are being analyzed, then only the desolvation terms are calculated. But this is done automatically within the software. So that's why I said during docking, it's not solvated. But when you're going for energy calculations, it is solvated and desolvation comes into the picture. Now, usually hydrophobic part of a ligand, we expect there should be an interaction, right? But it is now exposed to a water, and this particular phenyl part is very unhappy in the water, and it does not form any new hydrogen bonds or the like. Usually we expect being it's a phenyl, we will have some interactions, at least phenyl-centric hydrophobic interactions, but nothing happened because it's not happy in water solvated state. So when you're looking at delta G approximations, HYDE is the algorithm that is used in CSAR, which has a log P which connects to dehydration and hydrogen bond. Computation is fast, which is something similar to MMPBSA. If you're really much interested, you can look at this paper, which is published in 2013, where it talks about the delta G approximations. And the main for the pre-minimization, because after you have the complex, after the docking, only your complex gets generated. After that, you pre-minimize it. That's why I said it is similar to MMPBSA, but not as accurate as molecular dynamics. We check with the clashes and torsion energies. Only if clash and torsion strains are low, the values are meaningful, otherwise not. And that is a problem with Autodoc also. In certain interactions, you will be able to see there is an interaction happening among the atoms within the ligand, or there are interactions happening among the atoms within the amino acids. That is not acceptable. And the torsion energies are also sometimes unacceptable, which I'll be showing you in the next slides. So we also wanted to look at the contribution of the hydrogen bonds and also to log P with the desolvation terms. That is how we calculate the delta G. Now, this is something that is very important where we are trying to find out whether your compound achieved bioactive conformation or not. See, when you do a single compound docking, you will get multiple scores, right? And you choose the top one as the best one. But the question is whether that compound, that conformation is crystallizable or not. Are we checking it? No, we never check it. We might check with the torsions or anything, but that is a post-processing step after docking. But are we doing it? No. Many papers, they do report it, but we are not doing it. So that's what we have to look at. It's not about only scoring. It's about, we have to see whether the contribution of the atoms, how far it is to the affinity. When you see a bond, a dihedral bond for that after the pose is being generated, if it is green in color, it tells you that this particular dihedral or torsions are present in the Cambridge structural database. Cambridge structural database is the X-ray crystal structure database for small molecules. So in the backend, in the CSAR, they will try to compare between the torsions with the database. The database is not provided to you. It is a paid database, but its information is being embedded within the software. If it is an yellow color like this one, it says it is there in the CST, but not very much frequently observed. But if it is red color, it says it's rarely seen, so it is might not be crystallizable or not. So that just gives you a hint. Another thing is that when you want to do optimization, for example, you are taking a standard drug and you want to make modifications because your plan is to patent and come up with a novel drug or novel drug-like molecule. In that case, the software will be able to identify and tell you which atom is having more contribution to binding affinity and which atom is less contributing to binding affinity. Now these, whatever the color, the green and the red color circles, they are called as coronas. Of course, not coronavirus. So they are the circles or the balls surrounding the atoms. Anything in green color tells you they are of negative value energy. Anything of red color coronas, we tell you positive value energy. So technically it is said if delta G is positive, it is negatively contributing to your affinity. There is some issue with the structure, right? So if this particular carbon is having a 7.6 kilojoules, that is positive, right? And here we have the desolvation energy and the interaction energy for ligand and the for the receptor. Same way, for the negative valued binding energy atomic contribution, you have the desolvation terms as well as the interaction terms energy here. When you add up or sum up all of them, that is minus 3.4. So here it tries to solvate it, desolvate it, and consider the calculation. Now, when you sum up all the binding energy values of each of the atom, that's where the total binding energy that you will try to get. But again, I told you, right? It is all MMPBSA based. So it's not so straightforward, accurate when compared to molecular dynamics. Now, this is the thing I was telling you, a case study, which was done with Bayer. 45 lakhs compounds were being used. So FlexX is the algorithm it is in the command line again, only for FlexX. You can do docking in CSAR also, but even for drug discovery hackathon, they have used a FlexX in CDAC server in order to dock against binding DB. The binding DB is having almost 3.4 billion compounds, million compounds. And the file size of it was around 3.4 GB. I am talking about the SDF file size was 3.4 GB. FlexX software, which is in the command line, was able to do that 3.4 GB of your molecules, ligand database, and do the docking in multiple CPUs in parallel condition in CDAC servers. The same software is also embedded within the CISA, and it is also additionally given if you wanted to do a large jumbo docking if you want to. But while you're doing FlexX, as I said, virtual screening, only the scores are being reported, nothing else. The topmost scores, you filter them, choose the best 1,000 or 500 molecules for the next level of studies. So here what they did is, based on the score, they selected 2,200 molecules, and then they did MMPBSA studies because it's fast, right? It is not MDs based. And then they found 974 molecules. These 974 molecules had interactions with those key amino acids. And all the fitting, there was no overlapping, clashes, everything was neglected. And we found only 172 compounds that is purchasable, others all has to be synthesized. And then we did a biological assay, around 116 hits were there. Did we find 172 hits from the docking? No, only 116 hits were there. That's absolutely a good hit rate actually. So this is actually, that is the expectations from docking analysis. So from this virtual library, where you have 45 lakh compounds, you came down to 2,200. And at the end, you choose 116 compounds. That is how you calculate the enrichment factor when you're doing virtual screening. Now let's also look at the flipping and other importance also. The high detects the subtle binding mode differences. This is the PDB ID I have used, 1 and 2V. The experimental binding affinity, energy is minus 23 kilojoules per mole. right? And in physics terms, it's mostly just kilojoules, not kcal per mole. But yeah, we do report in kcal also. The right unit is kilojoules per mole in physics terms. Now, if you see the same pocket, the same compound is interacting with this amino acids, also with these amino acids. Let's see what is the difference here. So both we have the different poses, and this is how it looks in the 3D pocket. One interacts with this left region and the, on the right region. All of these are making interactions, and you can see they are both are best by scoring. But when the energy calculation came, you can see this gave almost very similar energy when compared to experimental. This also gave a negative 12, but not that great when compared to this particular pose. That's why I said when you're doing scoring, you might not identify the difference, but when you do the binding energy and affinity calculations, then only you'll identify the difference. The next striking analogy is scaffolding of fragment-based approaches, where I don't know how many of you know about PROTACs, PROTAC, right? E3 ligases and all. So here we try to find linkers between the two ligands. So we already have a module within CISA called as RECO, where it can do merging and linking. If you have two ligands at two different regions of the protein or two different proteins, you can find the linker between these two ligands which could bind. Or if you have a compound, you want to replace the middle or the nuclear region or the core, that's where the core replacement is taking place. Let's imagine your pocket is so big and your ligand 1 is very small, but you wanted to have some of the conjugates to be added. There we can do growing. This is the place where I told you, you need to ensure that these results are validated by an organic chemist to make sure these are synthesizable. So we just define where we want to make the replacement or scaffold replacement. And then we do that. And then we do a search 3D fragment library, and then we do replace it. So that's what we tried to do on the fragment-based library. And this was being with the collaboration with Roche and Matthias Rere from Biosolve IT. They have developed this technology. And then you have synthetic accessibility check, when you're doing, as I told you, software automatically takes care of checking whether this is possible to synthesize or not. For example, fragment creation. First of all, we wanted to understand where the compounds could be broken and where the fragments could be generated. And then we also tried to make fragments where a chemist could synthesize. And then we try to link the forbid, the unwanted bond formation. So we wanted to define all these based upon organic chemistry. Now, using pharmacophore of the receptor, again, I'm repeating, defining the pharmacophore constraints in the receptor, you will be able to identify the possible fragments from the database. So the software will be able to identify the possible fragments from the database, and then they merge them based upon score and affinity, and then they form a fragment-based compound. And Gleevec is something having a good history about fragment-based, and it is its marketed name is called imatinib. It's a very famous anti-cancer drug. If you ask me what is the success story of CAD on pharma, there are more than 112 plus molecules gone to clinical trials. I didn't say. You can check these two review of literature where they have mentioned most of this compounds, and also the link which talks about the fragment-based success stories and failures of fragment-based drug discovery. Don't worry about noting down this. All these slides are available in my slide share. I'll share the links at the end of the slide. Madam, could you please tell me what is our time limit?
We kept it for 2 hours, the morning session, then 1 hour break, and then 2 hours hands-on.
So I have another 15 minutes. Okay. Great.
Yeah, but you can extend 5-10 minutes. It's okay.
they shouldn't be over information with exhausted. So we have to give some.
I guess we all are enjoying.
I'm happy. Great. So, but think that all these drugs are with the assistance of CAD. at each and different regions. And don't think that these all has come to the market or to the clinic just by doing docking. No, there are plenty of things has to be done. Docking is just the beginning stage, maybe a 5% of identifying the hits. And then it comes for the optimization part. So don't think docking is drug discovery. No, there are more beyond that, which we will discuss. Now comes to one of the most important slide in this whole slide deck, the objectives of CAD. So here it's what I want to define 3 different terminologies: hit, lead, and drug. Can anyone tell me when we say, when do we call a compound to be drug? Either unmute and tell, or you can put it in the chat. When do we say a compound to be drug?
When it has been tested, you have done all the ADME assays, and it is tested and it is ready to go to the market.
Okay. Okay. When it benefits more than it harms. Okay. When available to market. Any other? It has passed with the criteria of admit. Okay. After NDA. Okay. If it inhibits or activates a cellular mechanism, it is considered as drug. When a compound is bound to target site and produces relevant results. Okay. So I can understand everybody is thinking and already know about when it is easily metabolized, best efficiency, when it queers. Okay. So I have some right answers here.
उसकी वजह से कितना स्टार्टअप को वेट खा रहा है वो.
Okay. Okay, fine. So the best technical answer is when to call a compound to be a drug is after NDA, basically when it passes clinical trials. Not ADME, not target site, not binding site, nothing like that. Once it passes clinical trial or when it goes to the phase 3, phase 4, after NDA, that's where we call it as drug. Why I want to ask you this question? I don't know how. I don't know how I am getting muted. I think you can hear me now. Okay. Yes, you can. Great. So, so that's where we are. Oh yeah, thank you. National Drug Authority, yes, NDA or drug approval. No, it's authority, right? Okay, fine. So yeah, you can hear. So basically, I put the title as drug discovery. But when I went for the Viva was, the examiner asked me, did you find a drug? I said, yes. Do you know what is the stage you can go for a drug? I was so confused there. So it is very sure that we are always looking at drug candidates. We are not looking at drug. The candidates are like job candidates. You are not an employee yet, you're a candidate. So what is a hit molecule now? Hit molecules are molecules that can tightly bind to your target, which has good potency. That's all is your hit molecule. We are looking at a micro level on a target site. But then what is a lead molecule? When you think that all your ADME and toxicity properties are being satisfiable or balanced, then we call them as, including potency, that where we call it as lead molecule. Right? Anurag, Gagandeep, I will answer to that question probably now itself. So you can freeze certain regions of the geometry of a protein. Please go through the tutorial where it has mentioned about freezing atoms or freezing regions of the proteins. And then those positions will be frozen and the rest of the region where you have modeled will be minimized, the backbone, basically. So there is a tutorial in NAMD and GROMACS. You will be able to do that for a specific domain or a portion of the protein. Okay, so great. So here, what I'm trying to say here, just a minute. Yeah, here what I'm trying to say here is that you have to identify a compound which is having optimal balance of all these properties. For example, a compound should be a hit. Once we find it as a hit molecule, then only we can look at whether it could be a lead molecule. Once, in order to say lead molecule, whether your compound is safe, whether it has good absorption rate in human intestine, what about the metabolic stability, whether the P450 metabolism, like whether it is whether your compound is forming a metabolite or it is stable, and what is the solubility and permeability of your compound. All those things has to be analyzed before you're going. So just predicting log S, log P, molecular weight, Lipinski rule doesn't mean that is ADME. ADME is more than that. Plasma protein binding category, then plasma glycoprotein, then hERG liability, cardiotoxicity, mutagenicity, many more. So don't think that few parameters, if you can predict, that is ADME. No. Now, in many of the papers, including me, earlier days, we used to just predict log P, log D. This has been predicted. This is good soluble, no toxicity. Okay, what is the use? Because ultimately, in order to call it as a lead molecule, I need all these properties to be balanced. When you predict these values, put it in a table and publish it, what is the use? You just predicted it. You didn't predict, software predicted it. But what is your conclusion from that? You will say that is solubility and toxicity. Is that enough for a drug? No, you need all to be at balanced. What kind of analysis that is being done there? I'll come up with a question there. So let's see what all interesting perspectives you have. So we wanted to quickly identify the situations when such things are not possible. So that's why I used to tell to my research students, come up with plan A, plan B, plan C. So that means when plan A is not working, if you have hiccup, no data, shift to plan B. But you should proactively know what you're going to do in plan B. And if plan B goes and if it doesn't work, go to plan C. Now I have intern students who joined 2 months back. They're already on plan D now because plan A, plan B, plan C didn't work. So that is how it is research, trial and error. Until unless we have the right data, we cannot work on. We don't want to simply always hypothesize things. We want to have prior good right data to have right predictions. So we need to have that. If that balance is not possible, change your track. You should be ready for that. It's not that you want to make it work means then it will be difficult. Fail fast, fail cheap. This is the answer for the question which I asked you from the beginning, how to reduce the cost of the medicine. If you think that it's not going to work, try to stop it at the very beginning itself. Don't try to dig it. Only when confident you move forward. Avoid missed opportunity. This is the where I asked you the question, how many of you use Lipinski rule of 5 before docking? If you do that, you will miss compounds. Don't do that. The reason is when you say Lipinski rule of 5, you all know that your molecular weight should be less than 500 kilodalton. How many of you will agree to me that 501 kilodalton compound is also a good compound equal to 500 kilodalton? So how can you avoid 501 kilodalton compound from your database? Right? So when you're doing Lipinski rule of 5, that is a mistake, right? I didn't say Lipinski rule of 5 is bad. I said the place or the step where you're using is wrong. Lipinski rule of 5 is absolutely used for oral bioavailability, but not for filtering compounds. That's a wrong step. When you're filtering compounds with, and in your data set, all the compounds will be less than 500. There are many FDA approved drugs which is having more than 500 kilodaltons. Did they do filtering there? No. So initial filtering shouldn't ever reduce your funnel size. Let docking decide it. After the docking, you better score, you choose from the virtual screening, but never ever do Lipinski rule of 5 initially. When you're going to do for natural compounds, for example, coconut databases, yes, there you have to apply some restrictions because certain natural products are very huge. And if your pocket is small, putting huge one on the docking doesn't make sense. So sound of science, relevance, very important. So understand the size of your pocket. Accordingly, give the size of your ligand from the database filtering, not Lipinski rule of 5, right? So keep that in mind. This side, yeah. Now I told you earlier, right? A chemist, when let's imagine that you have a compound A and B. Biologists do molecular level studies and said A is very good compound, B is moderately active compound. But then we go for toxicity studies, cytotoxicity, mutagenicity, cardiotoxicity studies, and we found that A is very toxic, even though A is active, but it is very toxic. B is non-toxic but less active. Now we are going back to a chemist. Can you help me to reduce the toxicity? Chemist will ask, what should I do to reduce the toxicity? Where should I make changes? If I make changes, the activity will go down. How will I make changes? So there are models which can help you understand which region of the molecule has to be reduced, right? This is through a tool called as StarDrop. So basically we can give you basically the evaluation for StarDrop. RLA does not have it, but I thought to tell you the importance of ADME also here. So that's how it is. Now, moving forward, there is another interesting question is that data overload is always a challenge. We have a lot of data generated from docking, QSAR, ADME properties, from physiochemical properties, all these properties. Now, how are we going to use this? That is a big challenge. So let me throw out a question here. We have 10 different compounds. Let's say we have 5 different compounds, and we have to carry out 10 different experiments. So 5 compounds, all these 5 compounds has to undergo 10 experiments. Each experiment, one is metabolic stability, one is cytotoxicity, one is bioassay, one is molecular level studies, one is solubility studies, one is each of the studies for drug discovery. All these studies are very important, or we can say all these studies are required to make a selection. Among these 5 compounds, I want to select only 2 compounds for my next level of studies. If I'm saying that using one single experiment, choose the best 2, it is very easy for me to do, because among that highest or the lowest, I will choose and go. But here the challenge here is I have to select all the 10 experiments data in order to choose the best 2 molecules. Do you have any idea how to do it? Good, you're thinking. How can we select the best 2 molecules among the 5, considering all 10 experiments data? This is the place where you have to think. You do docking, then you take that compound, you do bioassay studies. How the docking studies are related to bioassay studies? Can we relate it? I have seen in some papers they correlate energy with the affinity data. Please don't do that. Create a metascore based on significance of different tests. You are somewhere near Ayush. Yes. Any other perspectives? So these are the challenges that we face with our research data. We have many data. How are you going to tackle this for these assays? Maybe create a model to predict, but data limitations are there, I suppose. Can we use AI here? Also depends on the target site and the parameter that is important for this target. If it is a tropical drug, absorbable drug, the different absorption solubility rates needs to be given importance. Randomization. Gagan, you're also somewhere close to my answer, especially on the last line. Okay, great. Thanks for all your responses. I'll come to that answer. So as I said, we have a lot of data. We have docking data, in silico data, in vitro data, in vivo data, all data we have. Now we have to, very important step, we have to prioritize. As Gagan and Ayush and all mentioned, we have to understand which property is more important and which property is less important among the 10, right? And another less importance can be easily found out with uncertainty. I told you in the beginning, If your experiments are not repeated, and if you don't have a standard deviation or standard error value, then experimental data variability will be there. That is uncertain. We are not sure whether it is correct or not. So if we do not want to consider a property, even though if it is important, but if we don't have a valid data there, that should be given less importance. So in your prediction data, we don't want error data to be used. We want to use the right data. So first thing is choose the importance. Second is uncertainty has to be given less importance. Finally, selection. Selection means not simply clicking sort highest and then select. No. First rule is that when you're filtering first set of molecules, your quality should be given less percentage of weightage. More weightage should be given to diversity. Means you need to have different class of chemical compounds. That gives you enough room for optimization. That is very important, right? If you give only quality, that means only based on score, you are going to select compounds. There are possibilities you might fail at the end. When you have diversity, even one particular class of compound is failing, the other class might be there for optimization. And that can be done only manually. Don't click a button and sort it and then pick it up. No, you have to spend time. So this is how you have to apply data to guide decisions. Generally, what we do for prioritization, this is how usually we do. I think most of you will agree to me after once I finish the slide. First, you select some compounds based upon docking and all of the studies. You do potency studies, experimental, and then you remove some compounds. And then you select some compounds. Based on that, you do absorption rate studies. Again, you remove some compounds. Some of the good absorption rate studies, compounds will be selected for metabolic stability studies. Again, you remove some compounds which is not stable. Ultimately, you expect that this is your best compound that is coming. When I presented this to most of the customers, clients, and prospective pharma industry, they said this doesn't work. Almost 95%, this is what it happens. Even though your compound is very active and potent, if the absorption rate is less, it might get out of your threshold. That means missed opportunity. You might end up with a not that great active compound. So you have to have a decision making how these all properties can be considered together to choose the compounds. This is exactly you're doing Lipinski rule of 5. When you say, I want only compounds less than 500, your good compound might be going out because it might be 501 molecular weight. Don't do that. That's why I said, don't do that filtering initially. So you have to be very cautious with this kind of strategy of method. That's where you are looking at uncertainty, which I already told you 2 times. I'm not going to repeat it here. But when you're coming to statistical uncertainty in prediction, even in docking, molecular dynamics, QSL, everywhere there is uncertainty. There is error, but you have to report it. Until unless I see a standard error reported from QSR, that particular paper will go for several revisions. If you're not reporting the error value, standard deviation for a ADMET prediction, that result cannot be accepted, which tells you the applicability domain. It's very, very important. So that's where we are looking at a multi-parameter optimization. The same Lipinski rule of 5, similar rules are being applied, but we also add an importance level. What is the significance of this importance? Let me tell you the example here. Ligand efficiency, I said it should be greater than 0.3. I will say any compound greater than 0.3 will be given a score of 0 to 1. So the score is from 0 to 1 is the best one. Anything greater than 0.3, I will give the score as 1. Anything less than 0.3, between 0.1 and 0.3, I will give a score of 0.25. And I will say anything less than 0, ligand efficiency, score is 0. The advantage here is that I'm still considering the ligand efficiency, which is less than 0.3 and 0.1 with less importance. So I'm not neglecting completely. When I'm looking at molecular weight, let's say less than 500, I'm saying the score will be 1. But this molecular weight from 501 to 550, 50, I'm making a score as 0.2 or 0.3. Still, I'm considering that compounds. So that is called don't miss opportunities. Still consider them. Later, we will remove them at the end of the stage. We need to have our funnel a good amount of pool of compounds to filter at the end. Don't filter at the beginning itself so that you will miss opportunities. So this is already being proven in CNS drugs. This is already published in 2010, 11 years back. Have we applied it here still? No. So that is what already things are being there, but we are digging the existing knowledge. That's what I started my presentation with. We are still exploring the existing knowledge and going behind it. But there are many other things we have unexplored still. And QSR, one of the dangerous method, which I told you, we extract the data from the structures. We also take the response variable from experimental assays. We generate models using statistical and machine learning models. Here, I want to tell you, please don't look at only about R squared. R squared does not have any great significance when it comes to biology. R square is required. I don't say no. For statistical validation, you require R square. But keep in mind, if your molecular descriptors does not have any physical significance to the biological activity that you're predicting, the QSAR does not have any meaning, especially for regression. Regression always has the issue of overfitting and false positives. That's why we always recommend people to use partial least squares, then Gaussian process regression machine learning models. And also, whenever you are reporting your QSAR, ensure that you also report the error value, and then you have to validate it and predict it. And when you're looking at bioavailability, you're looking at absorption, liver metabolism, permeability, lipophilicity, as well as hydrogen bonding. Similarly, when you're looking at ADME, we are looking at A is for absorption, where will it get in through your human intestine. And then for D, it is distribution, where will it go after getting absorbed. And M is for metabolism, will it remain intact until it reaches the target. And excretion or accumulation, will it stay in. So this is what we are trying to achieve from ADME. Now you might ask a question, so just from the structures, how can we calculate the ADME? That is again SAR, structure activity relationship. So there are physiochemical properties that could be related to understand how far they are correlated with these ADME properties. And these are the main ADME properties. It's not only log S, log P, or log D. It's way beyond that, including blood-brain barrier. HERG is something very important. If you're patenting some compounds, then ensure that HERG assays are being done. Otherwise, pharma industry is not at all interested in your compound. Human intestinal absorption category, plasma glycoprotein, that is PGP, 2D6 and 2C9 are the isoforms of P450, then protein, plasma protein binding category, and all those things. Now there's and also thermodynamic properties for the drug delivery. There is another good question comes always from most of the participants is that, sir, there are Swiss ADME, PKCSM, paid softwares ABC. I use the same compound, predict with all these softwares, all are giving me different result. Which one I should consider? Should I consider the paid one, or the most highest paid one, or the free ones? The simple and single answer is the software which is telling you the standard deviation. And if the deviation value is lesser, that is more reliable data. What that means, let me explain. I'm just skipping that to, let me see if I have that just to explain it, because it's already lunchtime. I don't want you to keep you waiting. Yeah, this is the slide to explain that. It is about applicability domain. You have to understand whether your training set, this is all your molecules in the training set to predict solubility or toxicity, or let it be anything. That is a domain of applicability. You're bringing in a compound that is, let's say this orange star is your compound. If the structure and the predicted values are within the domain of applicability, the standard deviation will be less for this molecule. If your compound that you're bringing to predict is slightly away from the domain of applicability, standard deviation will be slightly higher, but still it is reliable. But if your compound is much away from the applicability domain and it is not having a very high standard deviation, it is unreliable. That doesn't mean that the model is bad, but your compound chemical class is not present in the training set. That's why the standard deviation is more. And that is where you decide whether to take which prediction. So standard deviation less, the better predictive capability. And there are different strategies. Let me quickly go through. So if you want to see whether there is a compound is metabolically stable or not, you can look at the regioselectivity, and you will be able to understand, the software will be able to say that this region is regioselective, so it can break down. So you can see it has broken down and form a keto group. And then there is something called direct nexus for human toxicity endpoint. These are not animal toxicity. These are human toxicity endpoints from the Pharma Consortium. And this is from LASA Limited from UK. So they have around 43 endpoints from human toxicity endpoints. These are all knowledge based. So basically, if there is epoxide present, they say it is mutagenic. So based on the presence of the specific structure, they will tell you which is toxic and non-toxic. And in fact, this report is very much mandatory for FDA when you're filing the compounds. Nova Bioster is again for, it's like a scaffold hoping and transformations that can be done. This is what you're linking the communication between a chemist and a biologist. You did some docking with this compound, which is with the replacement of pyrimidine. You got a better activity. Now you want to want to ask to a chemist to synthesize. He or she might say that not synthesizable, but you can see at the bottom, the software will be giving you the relevant references in order how to convert or transform lactam to pyrimidine. And there are also optimization tools using CSAR. You can use design new molecules. Of course, 2 o'clock we will be doing a workshop there. And reaction mechanism, bioisostere replacements, pro-drug strategy under the metabolism, and toxicity. If you want to reduce the toxicity, then the modeler wants to understand whether the compound that he or she has designed is synthesizable or not. So looking at the synthetic feasibility and assessment, this I already explained. And also looking at a case study here. Quickly, I'll go through this case study where we have around 3,100 compounds. We did a in silico ADME profile, and then we did a multi-parameter optimization from the scoring from 0 to 1. And based upon diversity and score, we selected only 10%. And then we did in vitro potency studies, and we clubbed the potency study and ADME studies together. But now we gave more importance to score and diversity in order to choose final 25 compounds. I always recommend you, whenever you are using in silico studies, to choose compounds for your wet lab studies. Don't end up with 1 or 2 molecules. That is all at the last stage. When you're doing docking or ADMET profile, at least have 10 or 20 molecules with you to do testing. I know funding limitations, but when you apply for funding, proactively shoot up your budget so that you show there for 40 or 50 molecules. They might limit your budget and you can end up with 10 or 20 molecules to test. Don't ever test with one less than 5 or 10. 10 is minimum recommended. So these things I'm skipping because it's all more of pharmacophore, which we are not doing today at the afternoon. But these are different technologies that is being already applied with the docking. And these are the different file formats that is supported, which is mol file, mol2 file, SD, SDF, PDB files, and smile strings are also used. Today we will see at the afternoon session. So that's all from me for today. We also work closely with many other programs for material science, as well as for co-crystal ligand, pharmaceutics, pharmacology, bioinformatics, molecular docking, and many others. And I take this opportunity to to thank all the people who have been interacting very nicely, also others who are patiently listening and also raising up the questions. But before we end up, let me show you one more thing, which is very important, which I was telling you. So people who are interested to learn basically your molecular dynamics, GROMAX, and you said if you don't have a good computer, you have only Windows, you don't have Linux, You can use this. Let me try to share the links in the, just a minute. Yeah. So this is a Google Colab. It will not take any of the resources or anything from your computer. It takes everything, GPU, everything from the Google Colab. It will take a graphics card also. It uses Tesla T4, which around 15 GB of the Tesla card. And you can learn, it will automatically install, run. You have to run it, that's all. It will run. So for learning purpose, you can use this. The problem here is every 12 hours, it will actually reset. Every 12 hours, it is going to reset your instances. That will be very difficult for you to run your research work here. So I'm using GROMACS 2021 version in GPU. So you can see this is a GPU code that we are running it. And let me see if I can show you the performance here. Just to show you how it ran. Yeah, let me check. Not here, maybe here, to just show you the performance. And I have taken matplotlib to plot the graph and everything. I can see many are being already using this. So just who wanted to learn, you can make use of that. Who are complaining that I don't have a good computer, or I'm not very good at Linux or something. So you can see the performance here. It is 126 nanoseconds per day. That is a performance here. But unfortunately, your real research, you cannot run every 12 hours. It deletes everything and you have to rerun it again. I have already a video for this in YouTube, which you can see how to be run. So let me post the link here. This is the link for the Colab. And then I am also working on the same Colab for NAMD. I'll be posting this soon. NAMD works for Windows also. Then if you want to download this notebooks, you can go to GitHub. Let me post the links here. These are all free tools, okay? So don't worry that sir is only talking about paid tools. No, anybody can use it. These are all freely available ones. For example, modeler notebooks also. For example, basic modeling as well as advanced modeling. It takes care of downloading the PDBs, it takes care of modeling, and it takes care of plotting the graph, comparison, everything is taken care on the go. Only thing you need to install is Anaconda in your software. Anaconda is a package with a lot of Python and other things that you need to install. That's fair enough for it to all things to work. Of course, you need to have modeler license also. So all the modeler scripts will be there, including all analysis and other things. Oh, sorry. Let me go to the end. Yeah, see alignment between the protein modeling, and you can also visualize the proteins using NGL view, and also graphs. Everything is there. Let me see. Yeah, I can see the graph. You can see the missing residues on the model, and I can also visualize this thing in the browser itself. And then we have a molecular dynamics using GROMACS. Already I have a GROMACS based notebook also similarly, which I have shown you earlier. And then today's slides, whatever you have seen, all the slides are here under this particular link. Let me again share that so that you have all resources with you. So today's slides is there. If you go to, there's the green, yeah, this one. Especially today's slides, role of CAD in MedChem. That's where all the slides today I have presented will be present. And there are many other slides talking about chemical databases for biological research, target identification using systems pharmacology or network pharmacology, then beginner's guide for molecular dynamics, and how to use applications of NIME and Anaconda in drug discovery, and many other details. Also ML, machine learning in drug discovery, all are there. If you want to see the demo on how to install Anaconda and how to run the GROMACS Colab, everything is there in the YouTube channel, which I will also put the links here. Okay, that's all from me for today. If you have any questions, I'm happy to take. Otherwise, we can continue with the question and answer session on at 2 p.m. since it is time for your lunch.
Thank you so much, Dr. Girinath Pillai, for such an elaborate and insightful session. and you answered all the questions very patiently and to the satisfaction of the participants. And yes, sure, we will take up if there are more questions in the session later on.
Sure.
So it's already 1:23. So can we get back at around 2:15?
Fine. For me, it's fine.
Okay. Thank you so much. Sunila, Dr. Sunila,
Yes, ma'am.
we can stop the streaming, YouTube streaming for the time being.
Yes, ma'am.